So if you don't know, this is Dr. Alex Mohr that's joined on the screen with me. He's a microbiologist, a scientist, has done a lot of really world-class publications, and has put together a test looking at the microbiome and its impact on health. So today we're going to be primarily focusing on his understanding of mycotoxins and mold toxicity in the gut. But just so you know, there's actually a lot more here. So we're just covering one small aspect of this relatively large report. And if you've seen some of the other webinars, you might have already seen this. I'm going to drop this in the chat so you guys can look through this. I'm also going to send you a link to the video that goes over this in a bit more depth. But as you can see here, we have quite a bit that the report itself covers. And so we were thinking it would be cool to do, to kind of break it down a little bit to where we're focusing in particular domains or particular subjects that are covered in this report. So today we're going to be primarily focused on the microbiome, which is this section here. And then we're going to be getting into resilience as well. So this might be a little bit of a newer concept for you guys, but very important to understand what resilience is, because it's going to change how you work with your patients and potentially change your perspective on them and how you're going to target with those patients in particular that have a high resilience.
We are going to go over a pretty complex topic. Of course, the microbiome, I'm sure everyone has heard about exactly what that is. Obviously, it's the collection of microorganisms that reside in our gastrointestinal tract. However, there's a very important subcomponent of the gut microbiome, which we term the gut mycobiome. Myco, not to be confused with micro. So I'm going to try to make that distinct as we go throughout the presentation. But really briefly, just kind of touching on the gut microbiome, again, harbored inside each and every one of us in our gastrointestinal tract resides a collection of microorganisms that increase in concentration as we descend down the colon. Now, based on the technology that's been available for approximately the last 2 decades, coming off the tailcoats of the Human Genome Project, we've had this vast array of technology, whether we're looking at different sequencing methods, bioinformatics, and now, of course, artificial intelligence that have really helped us decipher the gut microbiome. And the gut microbiome has been very attractive, whether we're talking about in the research world where I'm from, or translating a lot of that information for clinical application. I know there's many different tests now out on the market. We're going to briefly cover some of the technologies that a lot of these different testing reports and platforms are using. But basically, the gut microbiome is not only important for gastrointestinal health based on the proximity of where it resides, but also these new axes that we have formed through the investigations over the last several decades, including the gut-brain axis, this axis of communication that's occurring from the microbiome, whether we're looking at individual constituents of microbes entering into circulation, like endotoxins, or of course, their secondary metabolites, or which we term the metabolome, which a lot of that material is actually crossing over the brush border and coming into circulation and interacting with these tissues, like our brain, for instance. We know that it also, based on first pass circulation, is massively interacting with our liver. It can affect our adipose tissue, particularly when we talk about meta-inflamed states. Just how vast these interactions are, it's really a frontiers right now in science, and we're uncovering all different types of axes, even some of the research that I've done looking at the microbiome in its application for athletic performance. So the microbiome is really compelling for its deep connection to human health, and really something that, as we're going into new areas of the so-called omics space, whether we're looking at the genome, the metabolome, the proteome, et cetera, the microbiome is right up there with something that you're really going to want to get a good handle on when you're doing different profilings, particularly with issues that it might be hard to kind of walk back and understand where issues might be arising from. So just some really quick A&P. So when we talk about the gastrointestinal tract, or the alimentary canal, if we're talking about through the mouth to the anus, we're talking about 20 to 30 feet. So there's a lot of surface area in the structure, and there's a lot of different places where microbes can reside. Now, based on different factors, different gastrointestinal factors, most of the biomass is residing in the colon. And of course, that's where we're taking, when we're doing at-home testing, that's the actual sample type that we're taking through a fecal sample. Now, that's not to say that there's not other microbial populations further up We know that there are microorganisms residing in the small intestines and even the stomach, but generally we see the most biomass in the colon, and we want to kind of keep it that way. We don't want to have encroachment, particularly things like bacterial overgrowth, which we can get a good handle on with some of our testing. And then obviously there's other more traditional testing available to profile that. Now, because the gut microbiome is in the gut, we see it associated with a lot of different gastrointestinal associated issues, whether we're talking about IBS, ulcerative colitis, celiac disease, sometimes cancers, ulcers, and then of course, broadening out to other interactions with the host. This is really offering a wealth of information if you're interested in assessing someone's overall health, or what we term the holobiome, which is really including, the person, whether we're profiling their systems biology scaffold from the genome down to the metabolome, and then of course the microorganisms that reside on and in the person. Really, there's two kind of separate bins that you can put all that information. So the microbiome is principally tied to our health through the immune system. We know with gastrointestinal associated lymphatic tissue comprises about 70% of our body's immunity, whether we have specialized cells embedded in the brush border, or basically the gut barrier, and then other specialized lymphatic tissue that surrounds this whole tract. So it is a prime signaling agent for immunity and inflammation. Now, generally, when we think about the microbiome, our minds first go to bacteria. That makes sense if we're looking at a normal gut microbiome sample from a bowel movement that's comprising in a healthy person about 95% of relative abundance. However, it's important to note that there are other microbial kingdoms that are also important to profile. These, of course, include things like viruses. You can group bacteriophages or phages within that category. There is fungus, which we're going to be talking about today, which we generally term the gut microbiome. And there are other important factors like archaea, or the archaeome, which is also included in our report. So if you're interested in profiling not only the gut microbiome, but also the gut mycobiome, there are different ways that you can do this. There are kind of proxy methods. You can kind of get a handle on, taking a univariate approach or looking at single features. You can do some proteomic-based enzyme assays. ELISA kits, for instance, are widely used in this space. There, of course, are different immunoassays that you can do, imaging studies even. I know some people use breath tests as proxies, and there's sequencing-based approaches. So here we're looking at things like RNA-seq. I know some testing platforms will use quantitative polymerase chain reactions, qPCR. And of course, really the workhorse of sequencing the microbiome has been looking at a specific ribosomal RNA. It's cheap, it's good at annotating the type of microbes that are there down to the genus level, but unfortunately it really misses out on profiling at the species and strain level, and really in terms of accurately annotating functionality doesn't do such a hot job. Now, we can target specific regions of RNA. Specifically, if we're looking at fungi, for instance, we can target the internal transcribed spacer, the ITS region. However, the gold standard in this space has been, for the last several years, metagenomics. And metagenomics is very powerful based on the type of sequencing that we do, and I'll go over that in just a second. But what this provides us in terms of data output is not only the types of microbes that are there, whether we're looking at bacteria, viruses, fungi, or archaea, but also what they're actually doing, which is probably the most important thing that we can profile in the microbiome. Because if we're just looking at features, then we're just drawing associations. Well, this microbe might do this, but when we get at function, we can actually tell you what is occurring in that environment. So the type of sequencing that we specifically use for our testing platform, and the results that you get in your report, leverages what we term shotgun sequencing. So this is basically whole metagenome sequencing. A sample in, we remove all the other, digesta, liquid, whatever. We get the microbial biomass, we lyse the cells to get into the DNA, and then we break that DNA apart into these little short reads, hence the shotgun approach. We then basically sequence those short reads and reassemble them into whole genomes, which we can then annotate with either the taxonomy or the functionality. And the rebuilding of the whole genomes is really the computationally intensive part and why this process takes a little bit longer and is also more expensive than something like amplicon sequencing or qPCR. So we get all that data, and then we can start really looking at the functionality of the microbiome and informing the analytical domains that Micah was showing you earlier before I put up these slides. So just to kind of show you how diverse the microbiome is and what we might be getting in a sample. Fungi are the most abundant eukaryote. It's an estimated, if we just look out, throughout the entire world, there's between 2.2 to 3.8 million species. So very, very diverse, very, very ancient, intricately tied with all sorts of different types of environments. We can look at places beyond the gut microbiome, for instance, our own body. We know that there's an oral microbiome, there's a skin microbiome, there's a microbiome associated with our genitalia. Fungi is present in all of those places, but its most abundant concentration is in the gut microbiome. Mold, which I think is one of the topics that we're going to, Micah wanted to go over in this particular talk, is a structure that certain fungi can form. So if we think, just looking around environment, like that dust-like colored appearance that we see from mold is actually due to the formation of spores containing fungal secondary metabolites. And these spores are really the dispersal unit for fungi. Many molds can synthesize what we term mycotoxins as well as cytospores, which together with lytic enzymes inhibit the growth of competing microorganisms. So when we talk about being a conditioning agent in the gut microbiome, the microbiome, while very small in biomass, can have a massive impact on the overall architecture of our microbiome, putting us potentially in what we term a dysbiotic state. The reason that we're very interested in the microbiome and specifically mycotoxins is because of how pervasive they are in our food chain. So this can result due to the fungal infection of crops. We can, of course, eat them directly, whether we're eating produce or grains, but they can actually also be in our livestock because a lot of the feed that livestock are given does have certain levels of contamination. In fact, it's been estimated that when we look at the global food supply, approximately 60 to 80% of global agricultural commodities have some presence of mycotoxins. So in many ways, you can do all that you can to try to eliminate these. There is a potential that you are still going to be getting exposure. So that's important because these toxins pose significant health risks, whether we're talking about endocrine disruption, gastrointestinal inflammation, or the prime issue, immune suppression, which going back to just how involved the immune system is with the gastrointestinal tract and the microbiota, it's very important to do profiling in this domain. So there's just showing you some corn. You can see some mold growing on the corn, and corn is often used with livestock. So again, you can be reducing exposure through produce and grains, but even with livestock, even if you're on a carnivore diet, you might be getting exposure. Another really important vector, unfortunately, because I am an avid coffee drinker, we do get a lot of these toxins present in coffee, unfortunately. So just kind of looking at some very high level characteristics of the gut microbiome, you can see right away that in terms of the overall relative abundance, it's extremely low. It's in most healthy individuals, it's less than 1%. And if we go a step up in terms of phylogeny or the tree of life, we have different levels of the tree of life, right? We can maybe go from the kingdom level, go down to the class level, we can get to the genus level or genre, as I'm showing here, species, and then really the most granular would be strain. When we look at the most prevalent genre of fungi in the gut, really it's about 50 features. So it's not a lot, but obviously we can get a lot more diversification when we go down to the species and the strain level. And these 50 genera are associated to varying degrees with different really problematic human diseases, whether we're looking at things like hepatitis, cystic fibrosis, obviously very prevalent one, obesity, IBD. And when we profile the microbiome, we're seeing a lot of prominent features tied to these different health states. Now, just like bacterial colonization, fungal colonization can be a really great barometer for informing us on what health trajectory someone's going to be taking. And I'll show you some of that by a product of age in a second. Now, I had mentioned there was a lot of extrinsic factors that can shape the microbiome, principally being diet. Obviously, antibiotics and antifungals can help condition this environment. This is actually something that we are including in our test. We're not going to go over that today, but we do have an antimicrobial antifungal domain where we look at the principal genes that might be driving particularly things like resistance. And there are also intrinsic factors that are very important. For instance, age as well as sex. So these are important things that you want to consider when you're doing this type of profiling. So if we look at how the microbiome is formed as you age, up until very recently, it was assumed that the main inoculation event of microbes entering your gastrointestinal tract wasn't until you had entered the birth canal, if you went through a vaginal delivery, and you had exposure to the vaginal microbiome, and then you had population events occurring. However, we're now uncovering that event occurs before birth. So we actually see fungal species start assembling before you're even born, which is pretty amazing. However, really the prime event is occurring right after birth. And this is when the diversity of the microbiome is really starting to increase, as well as the abundance, as we can see there with that line graph to your right. Now, at age 1 to 2, this is really a critical window because we start shifting away from, if you're feeding breast milk or formula, we're starting to introduce solid foods. So now we're starting to see things like Saccharomyces cerevisiae become a lot more dominant. And this can indicate basically the prominent role for diet in shaping the microbiome. In fact, there was a study recently done in Finland on preterm infants that showed that administration of amoxicillin, for instance, led to an increased richness and diversification of the infant microbiome, suggesting that antibiotics might actually stunt the natural maturation process of the microbiome. This is a big issue, particularly with how much exposure that we get to a lot of these different medications, particularly earlier in life. It can have a lot of ramifications as we age. And as we age, generally we see the diversity of the microbiome starting to decrease as well as abundance. However,
I have a quick question on that, if that's okay.
Sure.
Sorry for interjecting, but just for my own curiosity about antibiotics, applied to young kids, is this, as far as what you're looking at, just the oral application of it, or would application into the ears for ear infections and things like that have an impact on the microbiome as well?
Spatially, it will have an impact, particularly with the skin microbiome. But when we're looking at the gut microbiome, we're principally interested in anything that's coming through oral route. So yeah, when we're doing our antimicrobial resistance panel, we're primarily focused on oral route antibiotics. If we branch out and start looking at, like things like the skin microbiome, the scalp microbiome, yeah, that can certainly have an effect. So as someone ages, particularly after 65 years, we start to see a overall shift. I mean, we look at broad ecological measures. Number one, diversity pretty much kind of flat lines. And then number two, we get an increase in abundance of the microbiome, which can be a big issue, particularly as someone might have decreases in immune functioning and some bacterial overgrowth events, particularly related to things like Candida. Now, in the microbiome itself, we know that the microbiome is not functioning independently. It's interacting with other kingdoms. Particularly, there's tremendous amounts of bacterial interactions. In fact, a very common probiotic that a lot of people probably have taken it, the people that are on this call, Lactobacillus rhamnosus, has actually been shown in vitro to help control Candida albicans and actually reduce virulence. And this is done by actually altering the metabolic environment produced by intestinal epithelial cells, which in turn create an environment that is amenable to Candida pathology, basically an invasion and growth in the gut. So here we see bacterial factors that can actually inhibit the growth of Candida, as well as other factors that are generally associated with different probiotics that exhibit bile salt hydrogenase that can interact with bile acids and help actually reduce some of the abundance of things like Candida. Conversely, there are also some bacteria, if you look to the depiction to your right, that can promote fungal colonization. For instance, the opportunistic pathogen, which unfortunately a lot of us in the West have, Bacterio theta iota micron, actually enhances Candida colonization through induction of mucin production. So normally, if you're to go into the lumen or the tube of the gastrointestinal tract, for instance, in the colon, we'll see in surrounding the brush border the brush border cells or the gut barrier mucus layer. Now, generally, we want to have a healthy mucus layer, but we don't want to have mucus overgrowth because mucus, unfortunately, can be a fuel source for Candida species, particularly offering carbon substrate for their proliferation. So, a really growing area in this space is not only looking at bacterial interactions, but also looking at viral interactions, particularly bacteriophages. We know that these can have a great impact on the bacterial community, which can then affect the microbiome community as well as host immunity. So, this is something that we are including in our tests, looking at the gut virome and bacteriophages and how we can actually in the future leverage that for targeted therapy. I know there's a lot of pharmaceutical companies that are interested in that as well. So, the actual interactions that we're detecting from your microbiome sample, that's of course one thing, but also, as I was alluding to with different environmental factors, particularly with food, are very, very important. We have a very detailed health survey that we have everyone take before they send in their sample, where we look at different environmental exposures, dietary intake, even gastrointestinal symptomology via the gastrointestinal symptomology rating scale, even the Bristol stool scale. All these things help inform us on what might be going on in that environment and what type of assailants might be coming in from environmental factors. We can see here a lot of foodborne fungi can be found in numerous food products like vegetables, fruit. Fermented dairy does have it. Meat, as I was saying, has it, as well as fermented beverages. And these can come into the microbiome and interact in deleterious ways, including biofilm formation. They can produce different types of antimicrobial metabolites, as well as quorum-sensing molecules, and also alter some very important substrate, particularly short-chain fatty acids. Like butyrate, for instance, short-chain fatty acid is the primary fuel for enterocytes or the brush border. So when you have decreases in butyrate, generally you're seeing an increase in gut permeability, which can be very problematic when we're trying to keep the contents of the microbiome in the gastrointestinal tract and not having components come out of that area and start interacting with the host at large. Also, tying deeply into inflammation. We know that there can be a lot of different, and we're not going to go deeply into the immunological aspects of this specifically because of how detailed it is, but there's a lot of communication occurring. So, to get a good handle on gut health and the microbiome in general, the first place that you want to start is looking very broadly to see if someone is in a dysbiotic state. So we're not going to go over this particular domain in depth, but we do provide a dysbiosis score in our report. So this is informed by over 8,000 different metagenomes or 8,000 samples from individuals across multiple continents, dozens of countries, and many, many different disease states. So here we can see where someone's microbiome configuration is falling in this reference, and then also the primary drivers. So if someone is in a dysbiotic state, what do we need to address to pull someone out of that state? Or conversely, if they're in a healthy state, what are the primary elements that are keeping them there that we want to continue to foster while targeting things that might be holding them back from a higher score? We also want to look at toxicity. Some molds and mycotoxins exhibit antimicrobial properties, so we want to selectively suppress those different populations while making sure we're not disrupting beneficial microbes and beneficial functionality. Also tied deeply into this and included in our report, we look at intestinal permeability, or more colloquially termed so-called leaky gut. Mycotoxins can damage tight junction proteins, so things like zonulin, for instance. Perhaps you've taken a test that included zonulin. This is basically a tight junction protein that keeps the cells, the brush border cells, adheres together. We don't want to have those pulled apart because then we have a pathway for encroachment of different metabolites and products like endotoxins from entering into circulation. We also want to have a good understanding of the inflammatory response. So we do have an inflammatory panel. And again, sorry to bring up all these other domains of the test, but you see how tied together a lot of these different domains are and why they were important to include in our test. Of course, immune system modulation tied to that, as well as metabolic and functional changes. We know that mold toxins in particular can interfere with nutrient transportation in the gut. So, in many cases, we may not be getting all we can get out of the nutrients that we're taking in. So if we're, taking a specialized diet or on expensive dietary supplements, that might actually be preventing the full utilization of those different interventions that you might be taking. And even more distally, if we look at implications for the gut-brain axis, toxin-induced dysbiosis and gut inflammation can produce neuroactive metabolites. If we're looking at modulation of things like tryptophan and serotonin, and this can even affect mental health and cognitive function. So we see it has very broad ramifications throughout our body. Now, very important to overall ecosystem health and the microbiome is this idea of resilience. So you might be familiar with, say, mental or behavioral resilience. If you have some type of traumatic event, how quickly are you able to kind of come back to your stable baseline event, or think of it as homeostasis? This is drawing from the biological principle of ecology resilience. So if we kind of think of a resilient state as the overall configuration of your microbiome being stable, or if it is perturbated by some assault, can quickly move back to baseline. And when we're assessing gut resilience, what we really look at is the anchoring elements of the microbiome that are holding someone in a stable state. For us, this helps us identify, first of all, what is keeping someone in a healthy state. Are they having a lot of short-chain fatty acid producing bacteria, for instance, that are really creating a conducive environment and a lot of cross-feeding reactions that can support a lot of commensal microbes? Or do we have someone in an unhealthy, resilient state, meaning they have a microbiome configured for pathology in the production of detrimental metabolites? Say we're producing a lot of TMAO, say we're overproducing things like acetate, which is another short-chain fatty acid, which overproduced can be an issue, or we're having an overproduction of mucus. Just because someone is in a resilient state does not mean necessarily that they're healthy. You can also be in a pathogenic state and have a highly resilient microbiome. This is a big issue in the microbiome space, and actually moving someone out of this state, and is not often addressed. We think that, if someone is in a dysbiotic state, we can just interject some broad probiotics or, slightly changed diet. Really what we have to target is those anchoring elements in this pathogenic state, and essentially remove the populations of those microbes to create a bottleneck event where we can then repopulate the microbiome with beneficial microbes to pull them out. So resilience is really a key to what we're doing, particularly when we're doing longitudinal profiling, seeing a shift out of this state. And I think when we talk about any type of omic profiling, it needs to be a consideration moving forward. Now, we look at, specifically looking at the microbiome, we look at the diversity, we look at how interactions are occurring with the epithelial layer, and how much inflammation this is producing when we look at functionality. So kind of going over.
Do you mind if I jump in here and share screen just to kind of recap what you were showing there real quick? So if you're not looking at your screen, I highly recommend looking at your screen, because I'm going to show you kind of recap in more layman's terms how to clinically apply this test. Essentially, when you sequence somebody's microbiome, we talked about like we're getting really deep reads, etc. What we're going to do, and I'm sorry, I choked on my own spit right before I started talking. So I have that in the back of my throat. Hopefully you can relate. Anyway, so the Microbiome Wellness Index, this is going to tell you if somebody's in a dysbiotic state or not. So if they're way up here in the green, their microbiome is doing really well. If they're down here, and I'm way oversimplifying this, if they're down here in the red, they're not doing so hot. And so what we're looking for is somebody in a dysbiotic state. And we're trying to see the range on which that exists. If they're in a dysbiotic state, or really regardless of where they sit on this, the next thing I'm going to go and look at is say, okay, what are the important microbes and the important things in this person's microbiome? Then I get to look into to see the primary populations of that microbiome to perhaps start to develop an idea for why they might be in a dysbiotic state. I can also run up here to a sun plot and look at the microbial populations there, the archaea, the bacteriophages, the fungi, etc. And so now I'm starting to develop a little bit of a picture of, are they dysbiotic? And what are the populations of this person's microbiome? And so that's where I'm starting to go with this. And then from there, I'm trying to understand how diverse is their microbiome, because that's going to tell me how well they can take an insult. And that's going to indicate to me also how well they're going to adapt to treatments that I give this person. Then I'm going to go to resilience. So now what this is telling me about this person is how well are they going to move, change, and shift their microbiome. So if they're up here and they're deep in the red and they have really high resilience, what does that mean? That means it's going to be really challenging to shift that person up to green. It doesn't mean it's impossible, just challenging, because their resilience is essentially trying to drag them back into the red. Same thing with somebody who's way up here in the green. If they have a high resilience, it's a really good thing. If they're already performing really well and they have a high resilience, that's exactly what we want. We want high resilience and a really healthy microbiome. And then from there, it's going to start to, so that's kind of the big overarching concept. Are they dysbiotic? What are the populations? And how can I move this person? How easy is it going to be to move this person? Then I'm starting to get down into more the clinical perspectives of what do I have to do to get this person to shift, either to optimize or to get out of this not so healthy microbiome. And that's when I'm starting to go into the archaeome and trying to understand that, the microbiome, to understand the impact of molds on this person. And so that's how we're looking at this, is you can kind of take the high level stuff and then really start to refine that and get into probiotics, pathogens, parasites, etc. So this is kind of the details of that high level picture. And then we're getting into the protocols. So the protocols, we develop out protocols for you. Obviously, as a clinician, you can do whatever you want. And we have some more things here, like how they're digesting, creating nutrients, the integrity of their, just further things that are going to build that picture. But then we get into protocols, and this is what Alex, I think, will go over in a second, but we develop out protocols that we think are going to be the most likely to shift this person the quickest way possible. So anyway, is that how you would describe this test in more layman terms? Alex, would you say that's relatively accurate?
Yeah, that's accurate. And thanks for airdropping in. And Micah, I sometimes can get a little bit deep with this, but I think it's important to showcase how we're trying to approach this. But you're right. I mean, in many ways, the report starts off very high level, and you can get as granular as you want as you go throughout the test, even down to the keystone features, at the very end of the test, which I don't think you showed when you were scrolling through. But yes, that first score is really kind of your anchor for better understanding the whole test. So I'm going to go back to my slides. Okay. So in terms of what our test offers, as Micah was just kind of showing you there, going through the pages, first of all, really important, because of the type of sequencing that we're using, we're getting down to the strain level. So this is the most granular level that you can get in a microbiome test. Moreover, we're joining that with the functionality of each one of these strains to really better understand just how the microbiome is going to impact your health. Micah was showing some ecology-associated features like diversity, which of course is very tied to things like resilience. We do perform pathway analyses that you saw with some of those network analyses that we have broken out by the archaeome, the microbiome, and the virome. As he was showing you, there are actually 15 analytical domains in the report. And really I guess if you want to call it the special sauce of this report is the personalized protocols, where we are leveraging artificial intelligence via digital twinning, which I'll go a little bit into in a few slides. So looking specifically at the microbiome section of the report, we look at the taxonomy. Okay, so what types of fungi are present in the sample, at what abundance, and then also what key features are tied to those. And we provide scientist notes at the end of this section, giving you interpretation of this specific profiling. Very important, and of course, kind of the main point of this talk today is the domain or the subdomain of mycotoxin bioremediation capacity. So as I was alluding to, we have this broad exposure, and we can try to eliminate it as much as possible, but it's going to probably be omnipresent, meaning it's going to be something very hard to completely avoid. So for us, the inclusion of looking at the functional ability for your microbiome to essentially control this assault from different mycotoxins was a no-brainer. So here in this report, we see different functionalities related to different classes of mycotoxins and how well your body or how well your microbiome is doing at essentially detoxifying these different assaults. So we pull specific enzyme commission, or basically enzyme classes, that are related to bioremediation, and we show you where you're falling in reference. So we might be doing really well in, say, quelling aflatoxin aflatoxins, but you might be doing very poorly in other areas. So based on these results, we can tell you how, through interventions, to shift this to make sure that you're handling mycotoxin exposure very well. And then also, very important, how to essentially eliminate, if you are having issue with proliferation of different fungi that are contributing to this, and also, limiting exposure from different environmental factors. Tied to this, because probiotics do have targeted probiotics, do have application in this space, as I was talking about a few slides, we do a full profile of the probiotics that are not only present in your microbiome, but that you're going to be more amenable to grafting in your microbiome, because we know that not everyone is going to respond to different probiotics the same. So having a personalized protocol, whether we're talking about the specific strains to take or the specific amounts, like in colony forming units that you will generally see on a good probiotic, we make those specific recommendations. Moreover, also tied into these other panels, we have a full overlay here with the pathogen and parasites that could be present. So again, another important layer when we talk about particularly what might be driving someone in a dysbiotic state, as well as a full antimicrobial resistance panel. So looking at what genes are being turned on in your microbiome, whether it's due to past antibiotic exposure or other issues that might be problematic in the future, and making recommendations, not antibiotic based necessarily, but also some more traditional or supplemental methods that could be workarounds if you are having issues in this domain. So really briefly, to go over the idea of digital twinning, this idea or this type of analysis was actually taken from engineering. It was NASA scientists that were looking for a way that they could simulate different conditions on different aerospace materials, because it's very expensive to test those, obviously. So they created digital replicas of those materials and put them through many different simulations. We're doing the same thing with this data. We're creating a digital replica of your microbiome. We're using a vast catalog of different interventions, and we're basically having these multiple simulations on this profile to see what intervention is going to work best for which domain, and obviously overall scoring. So we can look at, for instance, the microbiome, and specifically looking at quelling things like mycotoxins, and then the microbiome, different pathogenic features at large. And based on this, you can really cut through all the BS, so to speak, and not make broad protocols, but makes bespoke protocols. So you can start moving your health state right away. This is presented in our personalized protocols, where we do make a recommendation on retesting. So this is when you can expect to see increases in your scores. The recommendation is based on domain and is provided a weighted score, where we basically look at the impact. Okay, how much is this really going to move the needle? We have a 5-tiered system, 5 being the highest, going down to 1. So really the 5s are really what you want to focus on. We know that compliance is always an issue with people. And if we give a unweighted laundry list of recommendations and they don't really know what to do, compliance can be a really big issue. So here, we're not saying that this is a la carte, and we're not saying you can just pick and choose, but we're providing a list of recommendations that you can incorporate into your own regimen to see increases in the different domain scores. So in terms of, if you're looking specifically at reducing issues with mycotoxins, our own curated library includes things like prebiotics, specific dietary fibers, obviously the probiotics that I was listing, as well as mycotoxin resistant diets. We know that there's specialized diets that can actually help with this, repairing tight junction integrity, specifically those proteins that help hold the brush border together, looking at different short chain fatty acid enhancing interventions, as well as fungal bioremediators, even bacterial bioremediators, how to combat oxidative stress from mycotoxins. In some cases, we may include mycotoxin binders. However, you do want to be careful with these, and we'll obviously have that in our report, because this can bind to other important nutrients. So this is not something that you want to be on continually. So I know I'm kind of running out of time here. I'm going through pretty quickly. However, say if we're taking someone and they have a particularly low score in the microbiome, different bespoke strategies that we might recommend, just to give you a snippet here. Principally, we're looking at reducing environmental exposure. So we're going to make recommendations on avoiding foods that are going to be prone to contamination. Obviously, altering the diet is going to be really important because everyone is eating on a daily, for the most part, everyone's eating on a daily basis. And this is one of the prime vectors of assault from mycotoxins. So diet is going to be huge. We can also have different types of detoxification and binding protocols. For instance, activated charcoal, bentonite clay. Hydration is very important. Specific supplementations down to the strain level and the actual amount, as well as prebiotics and different types of interventions that we know are very powerful at reducing fungal overgrowth, like caprylic acid, even natural remedies like oil of oregano. And then we always encourage different types of products that are going to boost up immune function, whether it's beta-glucans, curcumin, or other polyphenol-rich dietary strategies. So really simply put, it's as simple as you just collecting a sample in the comfort of your own home. Thankfully, we have specialized stabilizing chemistry in our kit that keeps your sample stable for 2 months at room temperature, but we recommend you get it back to us as quick as possible. We sequence and then we perform all the bioinformatics and digital twinning on your sample and provide you with a very comprehensive report, one that I think really stands out currently in the market. So with that, I will hand back over to...
Yeah, thanks for being here, guys. So I'm just going to show you a little bit more about the tests. I'm not going to do a lengthy pitch or anything, but again, just to reiterate, so if you didn't catch that the first time, this is what the sample report looks like. I have dropped this into the chat. We have a video on our YouTube channel that goes into the report in a more depth, going across each domain. It is in presentation format, but just to abridge that really, really quickly, we have a table of contents. So you have your conceptual summary, 15 key analytical domains, the protocol, and then the appendix, which is where you can find, the keystone microbes and whatnot. So they'll get an overall health score. That's going to give me just a very general idea of where they sit as far as, if they're a 58 and it's a person who's really trying to optimize, really trying to be healthy, maybe I want to get them up to a 65 over the next 3 months or what have you. And by the way, the microbiome is notoriously difficult to shift. It's not so easy as some other things. The metabolism, you see and not that it's easy, but you see a much faster shift within things like the metabolism. And then genetics, for that matter, genetics are great, but they aren't telling you the information that this is telling you. So this is telling you about where you currently are at, whereas genetics are just saying like, hey, this is what could be happening. And based on what could be happening, we think this is going to be good for you. Still good to sequence, but they're, apples and oranges. And so we as a company are building to where we have genetics, hormones and proteins. We have transcriptomics and, the metabolism and the microbiome just all in one spot. So that's what we're building towards. And we see value in each, but it's important to understand their distinctive features. The microbiome, you're going to be profiling that when, through your patient intake, or you're talking to a patient and they have gastrointestinal issues, right? If they are just doing really, really well in that regard on patient intake, maybe you look at something else like the metabolism or whatever you as a clinician, decide, right? But this is going to give you insight on that. So again, just conceptual summary. And this is going to be telling you like, hey, here's a visualization of what's going on in your gut. You're doing really good at mucus degradation, et cetera. And then going to, this is the microbes that exist within the gut into a sun plot, but we're going to be going to here to see if they're dysbiotic. And this is going to be really informing a lot because it's going to be telling us a lot about the person in terms of where they sit. If, they're doing a 2, but I want to get them to a 2.5, I'm going to start to learn here in a second what it's going to take to get that person there. So if they're in a very dysbiotic state, I might run up to the sun plot, or I might run down here to see what are the key features of the microbiome that could be causing that dysbiotic state from a high level. And then I'm going to be going down into diversity. Diversity, again, you want to understand this because it's going to tell you how much insult that person can potentially take, essentially. For example, and I'm not recommending antibiotics, but if somebody took antibiotics and they had really high diversity, they are much more likely to rebound from that and much more quickly. And then resilience is telling me how easy it is going to be to shift somebody out of their current state. So again, if they have really high resilience, regardless of where they sit on this map, it's going to be really difficult to change that person's ecology, their microbiome. And then again, we're just getting down into the specifics of things. There'll be biofilms that are going to be added to this panel. We'll do another webinar on biofilms going over that. But now I'm trying to understand what are the specific things that are going wrong with this person, or what is the low hanging fruit that I can resolve that's really going to help to drive the outcome I'm looking for. So we just touched on the microbiome. If they're in a dysbiotic state, it could be due to the microbiome. It could be due to something else. That's why you need all these different domains here. And we're just trying to add domains that are the most likely to be contributors to this person's current health state, whatever that is. And so that's why we're adding things like the biofilm index into this, and so that we can look into that and see what we might need to potentially do, virome, etc. Probiotics, I'm just going to run through this real quick. We do have pathogens and parasites. We do metagenomic sequencing, so we are able to detect parasites. The difference between metagenomic and like a biome is that you're getting a much more accurate picture of what's going on in the gut, whereas if you're using something like what Biome uses, 16S sequencing, you're getting a fairly limited view, and then you're inferring what it might be. So an example of an inference would be looking at the shadow of a building to try to get a figure of how big the building is, but you just want to measure the building itself. So we're kind of just measuring the building itself to get a figure on how big it is. Antimicrobial resistance might be informative to you, digestion, and then getting down to personalized protocols. As he mentioned, that's how we develop it, is through creating a digital twin.